Good morning. I'm very pleased and feel very privileged to be here today and be speaking at the Daylight Symposium. My name is Manuel Spitschan, and what I really want to do today is give you a little bit of the insight of the work that I've been trying to push forward in the last couple of years, trying to understand really what are the non-visual effects of light on physiology and behavior, how can we understand them in the lab, in the field, and how we can tie all of this together to really provide a solid scientific basis and evidence basis for optimizing light exposure in the built environment. And of course, I'm following here a talk from Tim Brown, who has laid a lot of the groundwork already in the principles that I'll get back to today. Now, what I really want to start thinking about is how we think about the non-visual effects of light in terms of different types of categories. And a lot of the work that I've been doing in the lab has really focused on understanding the biological capacity of our physiology in responding to the effects of light. And so typically then in laboratory conditions, what we can do is we can isolate specific conditions, we can generate light sources that have specific properties to allow us to tease apart what specific photoreceptor contributions are, and really create insights into the biological mechanisms that underlie the non-visual effects of light. And of course, that is a very, in a way, privileged evidence base, because a lot of the confounding variables that we might have when it comes to light exposure in the real world are somewhat removed or minimized. And I want to contrast this with, of course, the real-world relevance of these effects, where we are living in real buildings, we have specific lamps and light sources in our living room, in our offices, things that we cannot control, and contrasting this with the laboratory evidence and the biological capacity questions becomes quite tricky, because of course there's another element of variability, and that is behavior. Biological capacity answers things about, or questions about biological capacity, helps us understand what can the biological system encode, what are the outputs. Ultimately, if we're looking at the real world relevance, what we need to do is we need to understand how these things could be modified. Specifically, and this is something that I want to kind of try to bring into the field, specifically with respect to behavior. Because ultimately, we're not lab rats, we're not animals under constant conditions, we are people who make conscious decisions about light exposure, and also conscious decisions in a way about how we organize our day-to-day. And of course, these two things go really hand in hand. Questions about biological capacity and the real-world relevance cannot be separated. And I add another component to it, which is something that I've been really trying to think hard about over the last couple of years, which is thinking about recommendations, guidelines, and practice. And we've seen a beautiful example of how a scientific consensus can inform guidelines about minimum and maximum light exposure levels. And I want to push this even further, because there's another component there which we as scientists really need to be careful about, and that is how we communicate these results. As I said, biology is messy, behavior is messy and noisy, and so how do we distill the key findings that we find, both in lab and field, into the nuggets of knowledge that we can then bring to practice and bring into the real world? I want to zoom out a little bit, or zoom in a little bit into the questions of biological capacity and describe some of the methods that we've been using in the last couple of years. And of course, we start by understanding really the system, or the non-visual effects of light, from the perspective of what is the front end, what is the input, what are the photoreceptors in the retina that encode these things. And we know there's cones and rods that allow us to stimulate, or that allow us to see during the day, that allow us to navigate around in the world. And of course, only about 20 years ago, the intrinsically photosensitive retinal ganglion cells were discovered, which provide an alternative pathway, or an additional pathway, to encoding information about the visual environment. Of course, these things aren't really separated. We do know that cones and rods input into the IPRGCs. We also have evidence that IPRGCs might be influencing visual perception. But overall, we're thinking about these sort of 5 photoreceptor classes, 3 types of cones, rods, and melanopsin. Now, when you're, of course, exposed to a light, you're not just stimulating the melanopsin cells or just the cones. Any light entering the eye will elicit reactions in all of these photoreceptor classes. And this is why it's, of course, important to understand the impact of a given light exposure on all photoreceptor classes. And The CIE standard, the CIE SO26 standard that came out a couple of years ago, is exactly doing this. It is giving us a handle onto quantifying the effects of light on the different photoreceptor classes. Now, just maybe taking a step back, my background is really in vision science. I started out my career in experimental psychology trying to understand what is the visual system doing, what are the how do different cone photoreceptors, for example, contribute to color vision? And so, from that perspective, we know a lot about the visual system. We know how light and patterns of light specifically enable us to see color, motion, and spatial detail. And of course, the other interesting signal here is, or the other interesting system that we care about is the non-visual pathway, which is thought to be primarily mediated by the retinal hypothalamic pathway, which connects the retina, so the front end to the eye, to the hypothalamus. And there, the functions that are associated to this pathway are melatonin suppression, circadian phase shifting, alertness, and so on. In my research, one of the real questions, one of the core questions that I've been working on is how can we understand what individual photoreceptor classes are doing, and how can we develop techniques that allow us to tease apart the different photoreceptors and their roles. So I want to really zoom in here and get to the heart of a method that we've been working on, and that is quite a promising way to understand, okay, what are the different photoreceptors doing? Now, generally speaking, so I work with humans exclusively, and this is a relatively simplified view of how animal physiologists might approach this question, but generally, you might have different methods available if you're working in animal models. So one type of, or one class of method is one where you're removing cones and rods from the retina through genetic knockout methods or other methods, and then you're asking the question, which function is preserved when you remove the cones and rods? And so any function that you can then measure is very likely due to melanopsin. And of course, the flip side of this is generating animals that have no melanopsin or that have deficient melanopsin, asking, in the absence of melanopsin, what function can be supported only by the cones and rods. Of course, in humans, these genetic techniques are not generally available, and there's some genetic conditions in which some photoreceptors are selectively lost, but in the healthy human retina, we of course don't have this opportunity to look at, well, let's take a look at what happens when we remove one photoreceptor class, what functions are preserved. Coming back to the spectral sensitivities, because this really is the basis of the method that I just want to walk through today. It's the method of silent substitution. It has a long history in vision science, and my group and some others have been trying to bring this method really to try to understand how do different photoreceptor classes influence the non-visual effects of light. So we know the spectral sensitivities of the photoreceptors in the human retina are overlapping. They have a distinct spectral sensitivity, but the consequence of this is that if we took a single wavelength light, such as generated by a laser or an LED or broadband light that's filtered through an interference filter, this light will impact all photoreceptors, and that's because the signal that's generated in the photoreceptors is proportional to the weighted sum of the spectral sensitivity in the spectrum. And so as a consequence, we can't use single wavelength lights to really tease apart different photoreceptor inputs. What we can do, though, is rely on what's called the principle of univariance, which states that the output of a given photoreceptor, so the photoreceptor cannot distinguish between a wavelength change and a change in intensity, simply because what it's doing, it's integrating the spectrum, the incoming spectrum, with the spectral sensitivity. And there's infinitely many solutions that could produce the same, what we call scalar output. We can use this exact principle experimentally to create conditions in which we have two types of lights that are matched in one photoreceptor class, so invisible, effectively, if you were changing between the two lights, but stimulating another photoreceptor class. That's what's called the method of silent substitution. We have silent and stimulated photoreceptors. We can, of course, scale this up, and this is a method that we've used specifically to understand what are the melanopsin contributions to vision, what are the melanopsin contributions to pupil control, by generating effectively scenarios that are set up in such a way that we don't stimulate one photoreceptor class and we're stimulating another one. This can be done with various types of light sources. the maths underlying this basically tell us we need as many light sources as there are photoreceptors that we try to control, that we try to stimulate. Let me just walk you through how this works. We start with a background spectrum. Here it's a broadband spectrum that is generated by a xenon arc lamp. We increase the power near the melanopsin peak. That increases the signal available to the cones and melanopsin. And so what we basically need to do is find changes in the spectrum that then balance out these increases. And so this is a numerical optimization problem. How do we find then the settings on the lights that allow us to effectively create two spectra that are matched in one photoreceptor or one set of photoreceptor classes and stimulating another one or producing a positive or negative signal in another one? If you're working with light sources or spectral power distributions, this type of matched spectra is also called metameric lights. They are basically spectra that are matched in cone excitations, but producing a different signal on melanopsin. And this principle can be applied, of course, in vision. It can be applied in circadian photo reception as well. And there are some studies that have effectively exploited this principle to understand, okay, what is melanopsin? What do the photoreceptors do? I don't want to go too much into detail about the results. Just want to kind of orient you to this method, which I think is very exciting and very promising as we're driving towards understanding and teasing apart different photoreceptor classes and their contributions. I want to zoom out a little bit and go to the real-world relevance of all of this. And of course, when we're thinking about light exposure, one of the ways that we measure light exposure in the field is by using, for example, ActiWatches or sensors that are somewhat attached to either the collar or perhaps glass frames or spectacle frames, or some other way effectively to capture the light signal. And oftentimes what we do in the field is we take ActiWatches or actigraphy measurement devices and we measure light exposure through that. And of course, all of these measurements are great, but they're only approximations of the quantity that we really care about, which is the retinal illuminance. Ultimately, the biological effects of light are not mediated through the cornea, they are mediated through the retina. And the retina is receiving a version of the light that's reaching the cornea or that's reaching the eye, that is filtered through the lens and that is also modulated by the pupil size. So with a doctoral student in Basel, here's an experiment or a setup that we've developed, which is allowing us to measure pupil size as well as the spectrum reaching what we call the near corneal plane in the real world. This is an ambulatory setup. We have a lightweight eye tracker that people are wearing, and we are effectively measuring pupil size as people are moving through different types of situations. And what we can do here is we can then really measure the pupil size dependence of an individual as they are moving in different light conditions, thereby generating data that allows us to ask, well, how light sensitive might someone be? We're also very excited about a sensor that we have had the pleasure and privilege of working with, developed by the team of Björn Schrader and Janine Stampfli in Lucerne, which are corneal plane sensors truly attached to spectacle frames, and there's some really exciting work that hopefully will be coming out on that. Measuring real-world stimuli is one bit. We also need to recreate them, and of course, that requires specific technology. So another doctoral student of mine that I'm co-supervising, Ali Hexley at Oxford, has been developing methods to recreate photoreceptor stimulations. This is a paper that we published last year, really trying to push forward and augment our ways of reproducing color and light in controlled conditions. And Ali won a very exciting prize from the Optical Society about this. Just want to highlight a couple of other things that go in this direction of real-world light exposure. We've been working on developing a data standard that would help us compare different types of spectra or light time courses and activity data collected in different groups. We've also been working on a scalable, hopefully, light exposure behavior questionnaire, which allows us to assess what types of behaviors people are engaging in when they're exposing themselves to light. And all of these really strands of research really drive towards understanding, ultimately, the real-world relevance of these effects of light. Now, I want to briefly touch upon something that's been a project that's been really dear to me over the past couple of years, or couple of months, really, over the past year, which is communicating with the public. And of course, I mentioned, or I set the stage earlier saying, well, biology is messy, real-world exposure is messy. How do we communicate the findings that we generate to the public? And this is really something that I've worked on with a master's student of mine, Colleen Weinzepflin, who joined my group, and the pandemic hit, and we were wondering, well, how can we use her skills to nonetheless develop a great project for her? And so she happened to be a neuroscience student and an illustrator, and we combined these two abilities, these two skills, to develop a comic book which is freely available. It's on the biological clock, it's on light exposure, and it's about optimizing these. So if you're interested in this, please feel free to download it and share it. It's open access, so it can really be used freely. And importantly, we've also generated a number of translations through the dedicated help of some translators. So this is something where really we see the value in providing high-quality communication content and materials to drive forward this method of the importance of the non-visual effects of light. Now, to me at least, these are really three areas that I think as scientists, and not just as scientists working with other people, this is, to me at least, these are the core areas of how I see my work. It's understanding mechanisms, it's understanding behavior and real-world relevance, and it's also understanding how do we translate all of this work into the real world. Now, I am setting up a new lab, so if you're interested in working with me, I have various PhD and postdoc positions available. And with that, I want to thank you for listening. I'm really excited to be here again at the Daylight Symposium. Thank you.